Biology 107, Topic 20, Viruses and Other Self-Replicating Entities. So I'm sure you must be wondering what I mean by self-replicating entity. We're going to be talking about viruses. We're going to be talking about viruses a lot in this topic. This is a particular topic that is special to my heart, and I hope you enjoy it. I'm going to talk a tiny bit about this new COVID-19 SARS coronavirus 2, just a little bit. And if you have any questions, please do send me some questions, because I do have lots of answers and I know quite a bit about the topic. We're also going to talk about viroids and prions and transposons. So stay tuned at the end of the lecture to hear about prions and viroids and transposons. So first I want to start talking about herpes viruses. So usually if I have this in class, I ask if anybody here has herpes, and usually I get some chuckles, and nobody usually puts their hands up, but I am here today to tell you that all of you have herpes, at least in some form or another, and here I'm going to show you why. So usually when people talk about herpes, they're often talking about these viruses here, so herpes simplex 1 and herpes simplex 2. So if you take a look over here, herpes simplex 1 is usually associated with cold sores, although sometimes it causes genital herpes. Herpes simplex 2 is more commonly associated with genital herpes, although sometimes it causes cold sores. So what you must be thinking, well, if you take a look over here on the right, I have a number. So about 60% of the human population is thought to have cold sores, or the virus that causes cold sores. Even if you don't have symptoms, you may still be infected with the virus. If you don't have herpes simplex 1, you have about 10 to 20% chance of having herpes simplex 2, which causes genital herpes, even if you don't have symptoms. So that's not a huge percentage, but that is a lot of people on the planet. Certainly does not add up to 100%, but some of you will definitely have these particular viruses. So if you don't have herpes simplex 1 or herpes simplex 2, maybe you have another herpes virus. So what about these ones? Maybe you haven't heard of these ones here. The varicella zoster virus. This is the virus that causes chicken pox and shingles. So all of you now are probably young enough that you actually had the vaccine against this. This used to be a rite of passage. When I was young, about 95% of people would have chicken pox by the time they were 15, and some people would get it later in life. So you have a 95% chance of having antibodies against this particular virus. If not, also a chance that maybe you've had the Epstein-Barr virus. This causes infectious mononucleosis. You can see that number, 95% prevalence in the human population. That's about by the time you're 30. So if you haven't had it yet, there's a good chance you'll have it in your 20s. So 95% chance of having one of those viruses, 6% chance of herpes simplex 1. Even if you've escaped all those viruses, maybe you haven't escaped all of these viruses. So if you take a look, there are 4 other viruses that are herpes virus infections, and they infect 60, 100, 85, and 25% of the human population. Most of these ones are either very mild or asymptomatic, meaning you don't get any symptoms, but there's still a good chance you've had them. All herpes viruses, by the way, are infections for life. So if you have the virus, you will have that infection for life. So chances are all of you have at least 2, 3, or maybe all 8 of these. Scary. There's a very good chance that nobody here has this one. Notice it's fatal. This is actually a monkey's herpes virus, but it's fatal in humans. So before we really get into things, I want to share some virus facts with you. First is that we eat and breathe and we're exposed to billions of viruses on a daily basis. It is amazing how many viruses are out there, out in the wild, and our immune system deals with most of them on a daily basis. So take a look at this. This could represent a salad that you had for lunch today, and at one point that salad was out in a garden or a farm, and those vegetables had insects walking on them, and those insects had viruses of their own, and so maybe you have eaten a whole bunch of insect viruses today. Viruses infect every living thing. It turns out that wherever we look, we find viruses. And pretty much every organism on the planet that we've looked at has at least one virus that infects it. So they are out there and they are very numerous. Our guts, in fact, are loaded with E. coli, and E. coli has its own viruses that infect it. So here's the key point. The vast majority of viruses that infect us have little or no impact on our health. That's good news for us. Most viruses don't actually make us sick. So back to this slide. This goes way back to the very beginning of Biology 107. I showed you this slide. We were comparing eukaryotic cells. Here's an animal cell on the left. And bacterial cells. There's a prokaryotic cell on the right. And today I want to talk about viruses. Notice how teeny and tiny that virus is right here. That's actually an influenza virus. I'll blow it up for you. You can see it's a little bit bigger. Notice it also has a membrane and it has DNA or RNA. So we'll talk about virus structure in a minute. So what exactly is a virus? Here's a definition from a textbook. You can see it says it's a minuscule acellular infectious agent having one or several pieces of nucleic acid. So we'll talk about what that means in a minute. What does minuscule mean? It means very, very, very small. So let's compare if we just look at the number of genes of a human versus a bacteria versus a virus. So humans have somewhere in the order of 20,000 genes, and we're very complex organisms. Bacteria such as E. coli, much, much less, 4,000 to 5,000 genes is pretty typical. Viruses can be very small. They can have 10 genes. There's one virus out there with 3 genes. Some of them only have a couple dozen genes. So it's very, very typical for them to be very, very small in terms of the amount of information they have. So viruses come in all shapes and sizes. Take a look at this. There's a red blood cell, so this is very small, and there's E. coli for comparison next to that red blood cell. Let's blow that up, and if you take a look, there's E. coli and some different viruses right beside it. We'll zoom in on that even further. So there's some different viruses. We have the smallpox virus here. We've got some very tiny polioviruses. In fact, the poliovirus is almost the same size as the bacterial ribosomes. Many shapes and sizes. So let's take a closer look inside. So here's the anatomy of a virus. A virus will have some sort of genetic material. Notice it can be DNA or RNA. It doesn't have to be one or the other. It could actually be single-stranded or double-stranded as well. We'll talk about that. The DNA or RNA is coated by a protein, so it has a protein coat called a capsid. So the capsid is a protective layer around the genetic material. And this is it actually for a lot of viruses. A lot of viruses only have these two components and that's it. Now some viruses have a third component, we call it an envelope. So what is an envelope? It's really just a membrane that's taken when the virus escapes from the host cell, it takes a little bit of membrane with it. That membrane will often have glycoproteins in there. They're called viral glycoproteins. Sometimes you hear them called spike proteins because they look like little spikes under an electron micrograph. So let's take a look at some different types of viruses. Like I said, they come in many shapes and sizes. Here's a helical virus. You can see this has some sort of nucleic acid, and it's surrounded by a capsid wrapped kind of in a spiral package. Here's a couple of examples, Ebola virus and the tobacco mosaic virus. The Ebola virus, in fact, tends to be very long and spaghetti-like and will actually sometimes form knots on itself because it's very long and strand-like. Many viruses are polyhedral. So what do I mean by polyhedral? It means they're geometric in shape. Often they have these capsid proteins that form these little kind of like triangles, and then the triangles lock together and form a roundish shape, and on the inside we have the DNA or RNA. Here's an example of a polyhedral virus. So this is an adenovirus here, also very similar to polioviruses. Adenoviruses are some of the viruses that actually cause cold-like symptoms typically. Many viruses are enveloped. So here, for example, is the HIV virus. So notice we have RNA on the inside. We have a capsid, so the capsid is this thing here. This has a second capsid, it's called a matrix. And then around that we have the membrane. And the membrane, notice GP120 there and GP41, those are glycoproteins, GP for glycoprotein. This new SARS virus, the CoV-2 virus, is also an envelope virus. There's a picture of it. And influenza is also an envelope virus. The good news about envelope viruses is that they are susceptible to hand sanitizer. So hand sanitizer will work against this new SARS-CoV-2 virus. Lastly, some viruses fall into the category of being called complex. So here's a bacteriophage. So bacteriophage, bacterial means bacteria, and phage means eating. So this means eater of bacteria. This is a virus that infects bacteria like E. coli. So if you take a look, we have some DNA and we have a capsid. So this part here is very similar to those polyhedral icosahedron viruses. And then we have some complex areas. So down here we have the sheath and tail, and these are used for attachment to the host cell. So sometimes they have extra features, and these are often called complex viruses. So here's a scanning electron micrograph of E. coli being infected by a bacteriophage, and it is not doing very well. So viruses come in many shapes and sizes. We've got helical, we've got polyhedral, we've got enveloped, and we've got complex viruses. All of them have these things in common. They all have some sort of nucleic acid, RNA or DNA. They all have a capsid. Some of them have envelopes, some of them may have extra features such as tails and things like that. So I want to go way back to Biology 107, Topic 1. One of the very first things we talked about in Topic 1 is this, properties of life. So if you take a look, here's the list. All the properties of life. Life has order, it reproduces, it grows, it uses energy, it responds to the environment, it has homeostasis, and it adapts from generation to generation. In that discussion, we had talked about how cells fulfill all those particular characteristics of life. Part of the discussion is, what about viruses? Are viruses alive? That's a very interesting question. Do viruses have order? Of course they do. We were just talking about a minute ago how viruses have capsids, and those capsids in an ordered way surround their nucleic acids and genomes, so they definitely have order. Do viruses reproduce? Also, yes, they do. Viruses don't reproduce on their own, however. They usually require a host. They can't just feed them food and have them grow. They actually require a cell to do the reproduction for them. Interestingly enough, there are some bacteria like chlamydia that also fit into this category, and most people believe chlamydia is alive. So I think viruses still get a check mark for that box. Same thing with the growth box. We'll leave it there. So what about energy metabolism? A big no here. There's my big no right there. Viruses don't make or produce or use ATP. Responding to the environment, homeostasis, same thing. Viruses don't usually respond to the environment. What about evolutionary adaptation? So you may know that viruses such as influenza do mutate. You need a new flu shot every year. So the answer is yes, viruses do adapt. Sometimes they change species that they prefer, like this new coronavirus. It was originally in bats, and now it has found its way into humans, and it's doing a very good job of adapting and growing in humans. So I'm going to switch gears now and talk a little bit about how viruses do what they do. So how do viruses grow? How do they take over cells, and how exactly are they replicating themselves? There's really a number of different strategies. I want to talk about three different types of virus life cycles. You can see this first one is called the lytic cycle, and you can also see that we're using a bacteriophage in this model. So a lot of information on viruses has been determined in bacteriophage, partly because they're very, very easy to study, and we don't need a level 3 lab or anything like that to study them. They're killing E. coli, they're not affecting human health. So the first step is the bacteriophage attaches. So those tail fibers we have here, these little tail fibers, they recognize something on the surface of E. coli, usually some sort of glycoprotein or carbohydrate or something like that, and they're going to recognize something on the surface of E. coli. The second stage is that the genetic material is injected. Now in the case of the bacteriophage, it actually has an enzyme that digests the cell wall peptidoglycan in E. coli, and it basically burrows a little hole there. You can see that little blue DNA. I'll just circle it here. There's the blue DNA. It's getting injected into the cell, Now in this case, in this particular bacteria, the virus is actually degrading the host DNA. This doesn't happen with all viruses, but it does in many cases. It's destroying the cell's DNA and totally taking over the cell. Total hijacking. Step 3 is synthesis of viral genomes and proteins. So this DNA has gotten into the cell, and DNA is, of course, instructions. So now the host transcription and translation machinery, so the RNA polymerases and the ribosomes, are being used by the virus genome. And what is it doing? It's making new viruses. So you can see in that picture there, you've got all those new virus particles, we've got genomes, we've got capsid proteins, and so on. So then it basically assembles. This is kind of like a self-assembly process in that these pieces recognize one another through complementary chemical charges, and it forms these new little virus particles. So remember over here, I had mentioned that the virus is making enzymes that degrade the host cell wall. So that's part of the last step, is that those enzymes that degrade the host cell wall actually allow this virus to break open the cell, and the new virus gets released. So there they go. So this is why this is called the lytic cycle. Lytic means breaking, like lysis, and these particular viruses are breaking open the host cell and being released. So for bacteriophages, it takes about 100 minutes. You go from one virus particle to about 200 virus particles. If this was a human cell, you can imagine it would kill the human cell and cause some sort of disease pathogenesis. So if you get cold sores, that's the virus killing your cells, and those cells are getting damaged, and you're getting a little inflammation and redness around the sore spot. So I encourage you to go take a look at this video here. It's very short, 40 seconds. It'll be on the course playlist, and it is showing a virus that is infecting E. coli. It's a really cool animation, worth it to check out. 40 seconds of your life, and it's kind of fun. So I want to talk about a second type of virus life cycle. This is called the lysogenic cycle. I'm not 100% sure what lysogenic means. It means something like sleeping. But take a look. Step number 1 is exactly identical. You've got this virus, it recognizes a host, it's injecting its genome, and the genome goes in. But step 2 is a little bit different. So step 2, we end up with the virus integrating itself into the host genome. So here you get something called a prophage, or if this was an animal virus, we'd call this a provirus. So there we go, provirus. And it basically becomes part of the host genome. So imagine this now. The host replicates, and it's got new DNA. You can see that bacterial cell is undergoing binary fission, and now you have 2 bacterial cells, and the virus becomes a part of it. So this goes on for quite some time until there's some sort of stimulus. So in the case of many viruses, one of the stimulus is cell starvation, or sometimes UV light. In the case of cold sores, sometimes it's UV light, or sometimes it's other viral infections that tell the virus time to repropagate itself. So the next step is called induction, and that is where the virus starts to produce virus particles. So let's take a look at that. So here goes, it gets induced, and of course, what do we have here? Right here we have some DNA, and that DNA is instructions, and the instructions say, hey, let's make more proteins, let's make more virus particles. So the second part is very much like the lytic cycle. You can see really it's just a stage where it's resting for a while, and it's resting or sleeping or hiding as part of the host genome. So if you do have cold sores, this is something that you may have happened to you, is you get cold sores, they go away for a while, and then eventually they start getting expressed again. So there's one other life cycle I want to look at. This is called a retrovirus cycle. So retro means backwards. So what are we doing backwards here? Turns out we're doing something called reverse transcription. So you can see I have that word reverse transcribed there. So let's take a look at how this works. An example of this kind of virus is the HIV virus, and we'll talk about HIV in a little bit more detail in the second half of this lecture. So first stage is the same. You have a virus, and it recognizes a host cell, and it injects its genome. So in this case here, though the genome very specifically is RNA. So I'm just going to label that this is an RNA genome. There we go. So it goes into the cell, and now we have that RNA. So there's my RNA, and the RNA is converted to that's a D, double-stranded DNA. So that is the backwards of transcription. Remember transcription, we go from DNA to RNA, and that's done by the enzyme RNA polymerase. In this case, we have RNA going to DNA, so it's reverse transcription. So that DNA, in this case, you can see is getting integrated into the host genome, just like we did with the lysogenic cycle, and now we have some sort of lifelong infection as well. So let's talk about animal viruses for a minute. What is different from an animal cell and a bacterial cell? Well, of course, animal cells have no cell wall. So believe it or not, this actually makes it easier for animal viruses to infect animal cells. Secondly, animal cells have a nucleus. So what's the significance of that? Well, it turns out that some viruses will replicate in the cytoplasm. Those are usually RNA viruses. And DNA viruses often have to get to the nucleus. So that's two membranes they have to get past in order to infect the cell properly. So many animal viruses are enveloped, many more than bacterial cells. And there seems to be quite a bit more diversity in genomes in animal viruses. So you can see here I've written single-stranded, double-stranded RNA, or DNA. So you can have double-stranded RNA, single-stranded DNA. Quite a variety of different genomes. So I encourage you to go take a break and take a look at this video here on YouTube. Again, this is 1 minute, and it's a nice brief introduction to animal virus life cycles. Go check it out now, 1 minute of your life. So here's a diagram from the textbook showing a typical animal virus life cycle. You can see up here we have a virus. This is an RNA virus, and RNA virus is going to recognize something on the host surface, and it's going to infect that cell. In this particular case, this particular virus is entering by endocytosis. So we talked about endocytosis way back in topic, topic 4? And in this case, rather than penetrating the cell directly, the cell is being fooled into thinking that this is food, and so it brings it in by endocytosis. So we have a viral genome here, RNA in this case. The genome is getting duplicated, and here's our messenger RNA. And our messenger RNA is going to be made in proteins, and those proteins are made at the ribosome. Some ribosomes are soluble, and some ribosomes are found in the ER. So those ribosomes in the ER are making glycoproteins. Those glycoproteins are put into vesicles. The vesicles end up on the cell surface, and so now we have all these glycoproteins on the surface of the cell. That virus gets away from the cell by a process called budding, and now here we have our new virus, and it's ready to go and infect another cell. So something that's important to know about viruses is that they have a specific host range. That means they have a very specific type of cell that they can infect. So some viruses infect only human cells, some infect very specific tissues. Maybe they infect the skin cells or the nerve cells or the white blood cells or something like that. Let's take a look at some examples. So HIV is very specific. HIV stands for human immunodeficiency virus. That means it infects human cells only in a very specific type of cells called CD4 T cells. These are a type of white blood cell. We'll talk about HIV at the end of this topic. There's a picture of HIV. It's infecting a T cell or a T lymphocyte. You can see those little pink things are the HIV virions that are budding away from that infected cell. We already talked about T4 bacteriophage, infects E. coli only, does not infect other bacteria. Influenza viruses, you can see, are a little bit more promiscuous. You can see they infect humans and pigs and birds. So there's a little bit of cross-species talk between these viruses. And rabies viruses are very good at infecting pretty much any mammal out there. Usually they're associated with carnivorous mammals, but they can infect other mammals as well. So I want to talk a little bit about virus classification now. So you may remember this diagram here. I showed you this diagram. We have our DNA, and the DNA can get replicated, and that's done by the DNA polymerase machinery. Then we have transcription by the RNA polymerase. Then we have translation by the ribosomes. Well, here's something. Viruses don't have cells. They don't need to follow all the rules. They can be a little bit more creative in terms of how they do these processes. So how do viruses do these processes? Well, it turns out there are 7 ways that viruses can do these processes, and these have been classified, and they're called the Baltimore classification system of viruses. The first type is a double-stranded DNA virus. So there's probably no surprises here. This is exactly how information is transmitted in your cells. So if you remember, double-stranded DNA gets made into messenger RNA by the RNA polymerase. The RNA gets made into proteins by the ribosome. No surprises there. But there are many other ways that viruses do these things. Type 2 are single-stranded DNA viruses. So if you see in this process, we've got single-stranded DNA, it's converted into double-stranded DNA, and then messenger RNA, and then proteins. But why use DNA at all, some viruses say? Let's just start off with RNA. So we have double-stranded RNA viruses. We also have single-stranded RNA viruses, so this is a plus strand. I'm not going to talk about plus and minus strands, but plus strand is basically the sense strand, and the minus strand is basically the template strand. Remember, we have 2 strands of DNA. Well, we can have either strand of RNA look like that. So single-strand RNA plus strand, single-strand RNA negative strand. We can also have this here. This is a retrovirus like HIV. And then a double-stranded DNA that can go through an RNA intermediate. So I don't expect you to know all 7 types of virus replication. What I do expect you to know is this key fact. All viruses must make messenger RNA, and there's a variety of methods that they can do so. They can be double-stranded or single-stranded. They can be RNA or DNA. Remember that fact. So I just want to show you some examples of some viruses. You may have heard of some of these. You may have not heard of most of them. It doesn't really matter. I don't expect you to know these examples, but just showing you some viruses from some of the different classes. So class 1, adenoviruses, herpesviruses are some good examples of these. Some of them are enveloped, some of them are not enveloped. Class 2, we have something called a parvovirus, causes a mild rash. Class 3 are reoviruses. This includes rotavirus, which causes diarrhea in infants. A big class are these plus-stranded, single-stranded RNA viruses. You can see right here, I wanted to point out that we have coronavirus. So our new virus that's been circulating in the human population is in fact an RNA virus, and it is enveloped. We have the minus single-stranded RNA, that includes the rabies virus, and then we have the retroviruses down here at the bottom. So viruses are classified usually by looking at the nucleic acid, but you can see there is a bit of disagreement. There's actually an international committee on the taxonomy of viruses. So if you don't like the way that viruses are classified, go join the committee. Maybe you can make a difference from the inside. Take a look at this chart. You can see obviously they're classifying viruses based on genomes, but sometimes we look at viruses based on what they infect. We talk about bacterial viruses or plant viruses, or monkey viruses or human viruses, and so on. Sometimes viruses are classified based on their shape, their helical, or their icosahedral, or so on. But the main way to know is that they are classified by their genome, because that tells us a little bit about their replication cycles. So I wanted to finish up this part of the lecture on viruses with a little quiz. So it says match the macromolecule with the virus components. So first one here is proteins. So you may remember that viruses have protein capsids, but over on the right there is no capsid. So let's take a careful look. So it turns out protein matches with enzymes. That's right, some viruses have enzymes that come along with them. Nucleic acids, hopefully that's an easy one. We're going to match that with genome. Lipids, so we're going to match that with envelope. So remember that envelope means the virus has a membrane, and that membrane is made out of phospholipids. Lastly, carbohydrates, that matches with glycoproteins. Lico is for carbohydrates. Question 2. Which of these components belongs to all viruses? All viruses have a genome, and that genome is going to be DNA or RNA. And all viruses have capsid proteins. All the other components are sometimes found and sometimes not. So this finishes part 1 of our lecture on viruses. Part 2 will be a little bit shorter. We're going to talk about HIV, we're going to talk about influenza, we're also going to be talking about those other self-replicating entities.